Hello everyone and welcome to CSIR Net Answer Key discussion. September 2022 exam. We are discussing about the life science key for morning session. The subject is Developmental Biology Unit 5. The question from part B, question ID 16. Which one of the following statement is incorrect? Transient rise in calcium ion is necessary for egg activation in mammals. Or it is saying sperm induces egg activation and does not involve calcium. So these two are contradictory, which means one of this should be incorrect. Generally in straight part bit question, that's how it is. In many organisms, eggs secrete diffusible molecules that attract and activate sperm. That's known as chemoattraction, so that the sperm goes towards the egg. Capacitated mammalian sperm can penetrate the cumulus and bind the zona pellucida. Zona pellucida is the outer layer of the plasma membrane in case of mammalian egg, and only the capacitated sperms that has already undergone the process of the physiological change called capacitation, only they will be able to undergo exocytosis so that to release their contents, and that way they can penetrate the outside layer of cells of cumulus layer of cells and penetrate the zona pellucida. So yes, this answer is right. Means this option is correct. This option is also correct. So as I said, one of this will be incorrect. Now, as we all know, fertilization is a process in which calcium is very much involved. After the fertilization, only the trigger happens to increase the calcium concentration inside the egg. And that's very much necessary for the slow block to polyspermy as well as the egg activation. So it says does not include means this is wrong. Transient rise, temporary, it's a temporary rise. So when the sperm is penetrating the zona pellucida, contents of it is released inside, and that triggers the receptor which initiates a signaling pathway. Phospholipase C is activated, which in turn activates inositol 3-phosphate, which breaks into two secondary messengers, that is IP3 and DAG, and IP3 goes and binds to ligand-gated channels so that the calcium can be released into the cytosol. Calcium is generally kept low in the cytosol. It is more in endoplasmic reticulum but less in cytosol. But upon this signaling, calcium will be released in the cytosol in waves. It doesn't increase at once, it increases in waves. This initiation of embryo development depends on a calcium increase in the egg, which is generally induced during fertilization. In many organisms, eggs secrete diffusible molecules that initiate, attract and activate sperm, as in sea urchin. During mammalian fertilization, so those are called as chemoattractants, the capacitated sperm penetrates the cumulus layer and then binds the zona pellucida with its plasma membrane intact. Question ID 17, that is also a part B question. This is based on axis formation. In which of the following developmental events the fate of maternal somatic cell, that means the somatic cell belonging to the maternal body, that is determined first. That fate is determined, which then determines the fate of developing embryo. Now we all know that in Drosophila only we have our maternal genes. We have two sets of genes, maternal genes and zygotic genes. Maternal already specifies the axis maternal molecules, and not only that, even the somatic cells of the maternal body is involved in the axis formation, especially the dorsal ventral. So if you look at the entire oogenesis part where it happens, the eggshell is responsible for differentiating the dorsal ventral regions of the embryo, and which reflect the polarities that are present within the follicle. This yellow color thing is the oocyte, but surrounding it we have many nurse cells, and at the periphery we have something called as follicle cells. Now the follicle cells only are what will give the dorsal ventral axis. So when the nucleus, initially the nucleus is present towards the posterior side. So these are posterior follicle cells. So with this, the posteriorization of the oocyte is done by the follicle cells, which is a part of the maternal somatic body. Oocyte is the one which is haploid, and which will be part of the embryo, but these follicle cells are part of the maternal body. So these give the signal so that the oocyte, this region, becomes pasteurized, and that's where the nanos protein, Oscar protein, they get restricted, because we know nanos, Oscar, they are all responsible for the pasteurization of the Drosophila embryo. And after that, the nucleus will be migrating towards the dorsal side, future dorsal side. So again here the nucleus will be giving some signal. Actually, the nucleus will be releasing some molecules which gives the signal, which is the Gherkin protein. So by the Gherkin protein, again this follicle cells becomes dorsalized. Since this side Gherkin protein is not there, this side becomes ventralized. So that's how the dorsal ventral axis is formed by the fate of the follicle cells. So first fate of follicle cell is decided, then they give signal to the oocyte, okay, this side can be the ventral side. Okay, specification of dorsoventral axis in Drosophila determines the maternal somatic cells, follicular cells first, which in turn determines the fate of the developing embryo. So while yes, these all play a role, primary organizer in amphibian embryo, and where the primary organizer will form will depend on cortical rotation, presence of the DSH molecule, all that is fine, but fate of maternal somatic body doesn't determine them. Same in C. elegans, anyways, vulva is a completely embryonic structure, so it is done completely in the embryo. And specification of micromere in case of sea urchin, again it's dependent on the position of beta-catenin, accumulation of beta-catenin, but then again it is in the embryonic body, not in the maternal body. So here option 2 is the right answer. Okay. Next question, part B, question ID 18. This is from plant development. Which one of the following floral mutants show the pattern sepals, petals, sepals repeated several times? ABC model of floral development comes almost all the time. What knowledge is required over here? What are these genes? Like which category, whether A, B or C, which category of genes they belong? And another is what combination gives rise to what all holes in the flower. So if you look over here, the class A genes are AP1 and AP2. These are the genes for floral development. AP3 and PI is class B gene, pistillata, and agamus is class C gene. Now what they give rise to is only A separately they give rise to sepals, A plus B gives rise to petals, only C gives rise to carpels, that is gynoecium, and B plus C gives rise to androecium, that is stamens. That's what is written over here. These are the floral holes, what layers will be formed. So both class A and class C genes are expressed along with class B. And A and C are antagonists. Like if C is not there, A will take its place. If A is not there, C will take its place. Okay. So over here they have asked what will give rise to sepals, petals, petals. Now sepals and petals both are produced by A and B. Nothing to do with C. But on the other hand, you are seeing that carpel and stamen is missing, which means C must have been mutated. Now if C is mutated, A takes his position. So these all become A. That means what? Here also instead of carpels, you get a combination of A plus B. So A plus, instead of stamens, you get a combination of A plus B. A plus B was giving rise to petals. So there, and here you have sepals. So you have sepals, petals, petals repeated several times. Okay. So if class C gene is mutated, A will express in place of C in the third hole, and B and C together forms the petal in the third hole. That is mutation in which gene? Agamus. So not only knowing part C is mutated is enough, you have to also know what is the part C gene. Which is the part C gene? Means which is the class C gene. It causes transformations in two adjacent holes. Petals develop in the third floral hole, and the cells that would normally develop into fourth, that is gynoecium, behave as if they constituted an agamous flower primordium. So the phenotype will be sepal, petal, petal. So as you can see here, if everything was wild type, then sepals, petals, B plus C is stamen, C itself is carmen. But if C, carpel, if C is not there, then A is only continuing. As you can see, it's taking the position. So you will have sepal, petal, petal. You can have sepal also or petal also. So this will be the mutation which observes in agamus. So correct answer is option 1. Next question is from cleavage divisions. It's a match the following, question number ID 19. What is the cleavage pattern and in what species it is observed? So very popular question, most of the time it comes. All you have to look is that you must know there are 2 types of cleavage division. Holoblastic means a complete cleavage, meroblastic means incomplete cleavage. Holoblastic happens in isolecithal and mesolecithal eggs, which contain equal distribution of yolk and moderate amount of yolk. Whereas meroblastic incomplete will have lot of amount of yolk. The position, if it is in periphery, it is discoidal. If it is in the center, if it is periphery, it is centrolecithal. If it is at one end, it will be discoidal. Now, holoblastic isolecithal also has lot of category: radial, bilateral, rotational, and spiral. You have to know which representative organisms have what type. Okay. So if you look at this particular cleavage pattern for holoblastic, isolecithal, radial is echinoderms, amphioxus, spiral annelids, mollusks, flatworms, bilateral tunicates. Bilateral means separate it into 2 halves. Radial means either in the vertical line or in the horizontal line, cleavage division. Spiral is 90-degree angle. So whatever will be the one division, at 90 degree the next division will be there. And rotational is the first division, whatever it is, the next division will be in different planes. If for this cell it is horizontal, in this cell it will be vertical. So that way it causes rotational. It happens in mammals and nematodes. Mesolecithal happens in amphibians, that is radial mesolecithal cleavage. Meroblastic, telolecithal, there also you have bilateral in cephalopod mollusk. Discoidal you will observe in fish, reptile and birds. And centrolecithal you will observe in most insects. So they have asked to match the following, as I just told you. Centrolecithal superficial, which is for insects. So see, anything you can match. Anyway, C over 4 here is exclusive. None of the options are 4. So 4th is the right answer here. Isolecithal bilateral is tunicates. Mesolecithal radial is amphibian. And telolecithal discoidal is birds. Easy, match the followings always. Now coming to part C questions. As previous year, previous paper, question paper, here also they have given limb development. Question number 75 ID. In mice, the genes encoding TBX5 is transcribed in limb fields of the forelimb, while the gene encoding islet1, TBX4 and PITX1, all these are expressed in presumptive hind limbs. So they have already given which gene is responsible for forming which part. Following statements are made about limb development in mouse. Loss of TBX5 gene results in complete failure of forelimb formation. So already they have given that yes, it is responsible for forelimb formation, TBX5. So if there is loss, obviously there will be loss in the forelimb formation. And if you just look at here, the gene encoding TBX5, it is transcribed in limb field of forelimbs. That's what I said. So if loss of TBX5 is said, then it results in complete failure of forelimb formation. Hind limb bud growth and initial patterning appears normal when TBX4 is knocked out. TBX4 is responsible for hind limb. So if it is initially knocked out, then there will be initial patterning will be normal. But if it is knocked out, then leg development will be arrested prematurely. Okay. And PITX is also for the formation of hind limb. So if you are misexpressing PITX in mouse forelimb, so it will cause its muscles, bones and tendons to develop, because that will give rise to the hind limb specification. When islet1 is inactivated specifically in the lateral plate mesoderm, hind limbs do not form, which is consistent with a role in hind limb initiation. Islet1 is responsible for hind limb formation. They have already given. So if it is inactivated, then how can hind limbs form? They will not form. So this is what happens in this segment. TBX5 gives rise to wing, that is forelimb, and in the hind limb region it gives rise to the leg, along with PITX1 also. So you have different mutants. If there is TBX5 mutation, then that this forelimb formation is not formed, hind limb is only formed. Okay. So according to that, limb bud, so as we saw that you have to find out which represents a combination of correct statements. So definitely A is correct, which means it will be either A and B or A and C. So C is also correct. C is not wrong. C is not correct because misexpression of pitex in forelimb ceases development of muscle, bones and tendons. It is the other way around. Misexpression of pitex will actually cause the development of muscle, bones and tendons. It is straight away from Developmental Biology by Gilbert book. So directly, instead of ceases, it will be causes. So this is wrong, which means option 1 is the right answer. Yes, it appears normal when TBX4 is knocked out, although leg development will be arrested prematurely. And so if islet 1 is inactivated, hind limbs still develop. No, they don't develop. They won't be functional. Okay. Question number 76. The following statements are made about mammalian development. Zygote is a totipotent stem cell. Yes, totipotent is correct, but stem cell, no, not really. Okay. Inner cell mass are said to be pluripotent, cells of inner cell mass. So yes, as we know that zygote is a totipotent property as it reserves the capability of forming embryonic and extraembryonic membranes. However, it is not a totipotent stem cell. Stem cell is something which renews itself with the formation of one daughter cell. That's why A is wrong. Now the blastocyst, the embryo divides into two layers of cells, two groups of cells. One are the inner cell mass, which includes the cells that give rise to fetus, all the embryonic parts. And another one is trophoectoderm, which is the outer layer. They will give rise to extraembryonic fate. Since ICM cannot form all cell types, because these will be extraembryonic membranes will be formed by trophoectoderm, ICM cannot form that. That's why we will say ICM, inner cell mass, is not totipotent, but it is pluripotent. Yes, it can give rise to most type of cells, but not all. So here they are saying that the cells of inner mass are said to be pluripotent. Yes, answer is right. So either B and A and B or B and C. Anyways, A is wrong. So correct answer is option 2. C is also right. 3 regulatory transcription factors, OCT4, NANOG and SOX2, help maintain pluripotency of the inner cell mass. Retention of pluripotency of inner cell mass depends on some transcription factors. So you can see the embryonic stem cells. What is helping it to self-renew? Yes, these transcription factors, OCT4, SOX2 and NANOG. Okay. So hence, statement C is correct. These factors are important in maintaining pluripotency. And the expression of the caudal type homeobox transcription factor, which is nothing but CDX2, it represses ES cells to self-renew. So if CDX2 increases, then it will go for, it will not maintain it in self-renewal form. NANOG and OCT are aberrantly expressed in outer cells of CDX2 null blastocysts, suggesting that CDX2 suppresses not only OCT4, but it also NANOG expression in the TE cells. TE cells have CDX expression, CDX2 expression, that is, it is aberrantly expressed in outer cells. So trophoectoderm will have CDX2 and it will not allow the OCT4 and NANOG to be present in TE. So that's why TE behaves differently. Okay. So CDX2 actually suppresses OCT4 and NANOG. That's why they are present only in the ICM, not in trophoectoderm. But what is last statement saying? CDX2 upregulates OCT4 NANOG. No, it suppresses it. That's why in trophoectoderm, OCT4 NANOG cannot be present. So option B is the right answer. Option 2 is the right answer. Next question, again amphibian and avian gastrulation development or development is compared over here. They have given two lists, similar question like last exam paper also. They have given two columns and different parts of the avian development, colar sickle, primary hypoblast, primitive grooves and Henson's node. And in column 2 they have given some parts which are analogous, means compared to amphibians. So which is what? Now colar sickle is nothing but local thickening of an epiblast formed at the posterior edge of area pellucida. If you look at the avian development, so you can see here this posterior end, some cells have started to migrate towards the anterior. So this side is the colar sickle. Okay. it is formed as a local thickening of epiblast. So what is written over here? Yes, Kohler sickle. It is the local thickening of epiblast formed at posterior edge of area pellucida. So A will be 3. Now A is 3 means it's exclusive. 3 is the right answer. But still, let's look at the others. Zona pellucida is the layer that remains as the cell surface, forming the epiblast, while other area pellucida cells have delaminated and migrated individually into subgerminal cavity to form the polyinvagination islands. So zona pellucida, yes, it forms the epiblast, but the other area pellucida cells, they will break from each other, delaminate, separate into 2 layers, and they come into the hypoblast layer to form some islands. So here some cells will be going through inside. So that's what is the process of gastrulation when it starts. Okay. So yes, primary hypoblast. It is the delaminated cells from epiblast forming polyinvagination islands. Cells at the surface converge to form the primitive streak, and a depression forms within the streak. So that depression which is formed, it is called as primitive groove. And that's the part through which migrating cells will pass into the blastocyst. So if you remember in amphibians, we had the dorsal lip of blastopore, means we have the blastopore through which also cells go inside. So primitive groove is compared with the amphibian blastopore. If you see in amphibia, this is the pore through which the cells have started to go inside. This is the blastopore. So cells have started gastrulation from here. And in avian, this is the primitive streak, and the depression which is called, that is primitive groove. So it's through the primitive groove that the cells go inside. You can see how the cells are going inside. Okay. So primitive groove is nothing but the compared to amphibian blastopore, and the tip of it is called as Henson's node. Okay. So Henson's node is the gastrulation center in the avian embryo. It is the homologue of the amphibian dorsal blastopore lip. The groove part is the blastopore itself, and the nearby area, that is Henson's node, it is compared with the lip of the blastopore. So yes, here Henson's node is equivalent to blastopore lip, and primary groove is homologous to amphibian blastopore. Correct answer is option 3. Next question, 78, regarding C. elegans. C. elegans embryo uses both autonomous and conditional modes of specification. So here also it is about the signaling, which is for the dorsal ventral axis specification. The signals from P1 blastomere instructs the EMS cell for further development. Now let's have a look at that, what is happening. We have the C. elegans embryo, which divides into 2 cells, that is AB cell and P1 cell. Now a P1 cell further divides into P2 cell and EMS cell. AB cell divides into ABP and ABA. But actually initially AB divides into 2 AB cells. It's the P2 cell that gives signal to the AB cell to become posterior. So what is the signal? APX1 is the signal name, GLP1 is the receptor name. But that's not part of the question here. The part of the question is P2 giving signal to EMS. Now EMS further divides into endoderm for E and mesoderm for MS. So how is the signaling done? By MOM2 signal released from P2. MOM2 binds with MOM5 receptor on EMS. So whichever side it will bind, that is this side, that side it will form endoderm by decreasing the expression of POP1 cells. Okay. So in endoderm, POP1 will be less. So if P2 cell is removed at early stage, then this POP1 down regulation will not happen, because MOM2 will not be there if P2 is not there. MOM5 signaling will also not receive. So POP1 will not be degraded, it will be same as MS. In MS, POP1 is present. It is not down regulated because signaling is given to the E side of the cell, not to the MS side, because P2 is in touch with this cell. Okay. So if P2 is removed initially, then EMS divides into 2 MS cells only. Instead of E, you will get 2 MS cells. Okay. And what is this message? MOM2 is nothing but a WNT type of signal. And MOM5 is a frizzled type of receptor. The signal for WNT signal is frizzled. Okay. So this down regulates the expression of POP1. Now if POP1 is not there, in embryo generally POP1 is supposed to be there. But if POP1 itself is not there, then MS also cannot express POP1. As it is because of signaling, endoderm will not have POP1, but now even MS will not have POP1. So by default, this MS will again become endoderm. Even without signaling, it will become endoderm. So based on that, if you see, the signal from P1 blastomere instruct EMS for its further development. No, it's not P1, it's actually the P2 which is giving the signal. In the absence of instructive signal, EMS cell will divide into two MS cell. Yes, because instructive signal is from P2, then EMS doesn't get the signal to form endoderm, it becomes MS. So B is right. In the absence of the POP1 signal, the prospective MS can take up EFET because anyways POP1 is not there. So how mesoderm will be specified? It takes up endodermal fate only. So C is also right. Yes, second option is the right answer. MOM2 protein from the blastomere, which is neighboring the EMS cell, instructs the dividing EMS blastomere to take up MS fate. No, it will be giving signal to take up endodermal fate. Whichever cell is in contact with P2, it gives signal to MS side cell to form the endoderm. So this is not correct. Okay, option 2 is the right answer. Question 79 about regeneration in Planaria. Generally they will ask for amphibians. This time they have broken the stereotype and given Planaria. The interactions that maintain polarity during Planaria regeneration is shown here. Following statements are given. Now you can see all over here the green part is ERK. This side is the WNT. Here WNT is expressed. And notum is this blue. So first of all, WNT is inhibiting ERK. So wherever WNT concentration is more, you will see ERK concentration has decreased. So what is our conclusion? WNT inhibits ERK. Okay. And we can see that notum, it is inhibiting WNT because wherever notum is inhibiting, the WNT is not able to express. Okay. But wherever notum is present, ERK is able to express. So based on that, some statements are given. Notum, it is nothing but inhibitor of WNT protein. So yes, here you can write down notum is also inhibitor of WNT. See, these you are not supposed to know. Yeah, this is not a very common question regarding Planaria regeneration. But whatever you need, it has already been given in the question. Now based on that only you have to answer. That's all. So this I already got from the diagram itself. Diagram they have provided in the question itself. Notum is upregulated in the anterior facing blastema. So this side wherever the notum expression will be there, the anterior facing blastema, you can see anterior means eyes are there, this side. So this side is the anterior. That means notum is expressed over there. It is upregulated over there. If notum expression is knocked down, now notum was inhibitor of WNT. So if it is knocked down, then WNT can produce. It becomes too much functionally active. Now wherever WNT was there, that side was supposed to become a tail. We can see head and tail, that also we can make out from the diagram. So in that case, what will happen? The anterior blastema, if notum is knocked down, it will form a tail. But what is written over here? When notum expression is knocked down, the anterior facing blastema will still form a head. How it will form a head? Notum is not there. So A is not correct. That means 1 is not correct, 4 is not correct. So C is definitely correct. When WNT pathway is blocked, the resulting Planaria will have heads on both the ends. Now that makes sense because WNT pathway, it was inhibiting the ERK. Now WNT is only not there. So then this will be present everywhere. Notum, right? So if the WNT pathway is blocked against either beta-catenin, then the posterior blastema also regenerates a head. So one will have two heads at both ends. Now we have to see B or D is correct. So when notum is expressed in posterior end, Planaria with two heads will be formed. So that's exactly what happened in this C case also. Because WNT is not there, so this side also notum expression was there, it will be head. So similarly in posterior end also, if notum is there, notum inhibits WNT. So because of notum presence, WNT cannot be there. So this side also head will develop. Okay. This result strongly suggests that anterior expression of notum functions to antagonize the posteriorly produced WNT, leading to head specification. So when notum is expressed in the posterior end, planaria with two heads will be formed. Also, if the WNT pathway is blocked, yes, this we already saw. Now, therefore, only in more anterior regions lacking WNT and can ERK induce head regeneration. If in the anterior region WNT is not there because of notum's presence, then only ERK can induce. But it's written high levels of ERK inhibit specific head specification. No, wherever notum is there, then only ERK can give. So this is also wrong. Correct answer is option 2, B and C. Last question in part C, developmental biology. It is again from plant. Given below are the list of proteins and their functions during floral induction. So you just need to know the different genes. Flowering locus C, FLC. It has a role in the timing of initiation of flowering. So it represses two genes that promote flowering, FT and SOC1. So because it represses this, it will affect the timing of the flowering. FT-FD complex is proposed to mediate transcriptional reprogramming of the shoot apical meristem by transcriptionally regulating flowering genes, that is your SOC1 and fruitful, which encode MADbox, all these transcription factors, and they will start a fluorescence meristem. So what is said over here? That flowering locus, what it will do, FLC, it has a strong repressor of flowering. Yes, it will repress the flowering genes. That's why it will be strong repressor. So A is 4 means 4th is the right answer. But still we will have a look at the others also. So FD, as I said, FD is mobile signal that induces flowering. FLC will, so FLC is actually repressing FT. Okay. Flowering locus FT encodes a systemic signal communicating the perception of long day photoperiod to induce the floral transition. And FR1 encodes a novel protein with two coiled coil domains that activates the expression of FLC. So FR1 will activate FLC, which in turn encodes the MADbox transcription factor that inhibits the floral inhibition. So FT and SOC, they will inhibit. So FR1 actually activates FLC. So FR1 frigida, it is an activator of FLC. D is the right. D goes to 1. And FT, as I said, it regulates target genes that mediate reprogramming of meristem to produce the floral inflorescence meristem. Okay, so option 4 is the right answer. So those were the questions. I would say the difficulty level was medium this time. But from many portions, questions were not given usual ones like vulva development, eye lens induction, and even the first subunit where terms and terminologies of development are given. And individual gastrulation development questions were also not there. Okay. So medium difficulty level. That's all, everyone. Thank you.